Josh, I've really enjoyed these conversations. They've been really intriguing to think about where we are now in the ADC space. And I think we're seeing huge changes from where we were in, as we described previously, version 1, version 2, version X of the ADC world. That's fine for where we are now and what we're doing right now. But where are we going? What's exciting you for where we might end up in the ADC space 10 years from now?
It's a good question. So if we look at the clinic at the moment, there's about 400 drugs in the market. Most of them take the same mechanism of action with payloads and linkers. Do you know what's one of the ones that's following up the closest? Greater antibody conjugates. And we're seeing that well from the small molecule building in the selectivity. We're now seeing that move into the large molecule world as well. I mean, you know more about degraders than what I do. It's not my specialist. So where do you sit on the field?
I mean, it's an interesting one, isn't it? And I think we touched on this previously. Let's take a big molecule with perhaps challenging PK, and then let's conjugate it to a beyond rule of 5 molecule with perhaps challenging PK. And yet you put the two together and you get this exquisite delivery mechanism to get your degrader exactly where you need it. And actually the overall properties are not that bad. We know they're deliverable. We know you can use them in the clinic. We know you can get these agents to where they're needed in terms of tissue penetration. One of the challenges with degraders is, particularly for something like a VHL-based degrader, is getting that oral bioavailability. But if we're dosing once a week IV in a clinical setting anyway, then that issue kind of goes away. So I'm really intrigued by the DACs, the degrader antibody conjugates, because I think they circumvent some of the issues that we're seeing with the with degraders, particularly with the heterobifunctionals, perhaps less so for molecular glues, certainly for the degraders. I think one of the areas I'm really excited about, and it's something I think we're just on the cusp of, is combination therapy ADCs. So the idea of dual payloads.
Yeah.
We see this time and time again as we see clinical data emerging. We know that patients with cancer become resistant very quickly to targeted therapies. And the only way to circumvent that is to dose 2 targeted therapies on different pathways. But we tend to dose at maximum tolerated dose. And the challenge with that is to get tolerability, you have to dose reduce one or the other of those targeted therapeutic agents to get to a point of tolerability. And by the time you've done that, you're below the biologically effective dose. I love this idea of dual payload antibody drug conjugates. So you can take those 2 molecules and deliver them to a patient in a targeted fashion, so you don't get all that nasty on-target mechanistic toxicity.
Yeah, absolutely. And we can always modulate the DAR of both of the payloads as well. So if we do have something that's more selective and targets a specific cancer pathway, let's say BTK is always a nice one because it's so prevalent within the industry. Let's say we have that kinase inhibitor on your antibody as a DAR6, and then you have a DAR2 of an auristatin. So we know we've got 2 mechanisms of action that are proven to not only kill the cell, but actually maybe move away from the resistance mechanisms that we can potentially see as well. We kill it fast enough, then we actually open up a new field and we're less likely to be resistant, and we get that higher therapeutic window and less off-target selectivity, as you say.
And I think one of the discussions that's ongoing at the moment is, well, do you take your 2 agents and put them on the same linker? So you have almost like a bifunctional linker, or do you engineer your antibody attachment points so you've got orthogonal attachment points so you can load up 2 different linkers with 2 different payloads, and then you can mediate that DAR ratio so that you get appropriate activity. But I'm also intrigued by the idea of being able to build in drug-induced synthetic lethality, if you like. We know that we've exploited synthetic lethality in oncology for a decade and a half now with the PARP inhibitors, but being able to load parallel inhibitors of 2 different pathways, so you get this intrinsic synthetic lethal approach. DNA repair inhibitors spring to mind. Or maybe we load up, as you say, a DNA damaging agent with a DNA repair inhibitor on the same ADC. I think the more you think about these new technologies, the more you see the potential for these approaches, taking that small molecule, learning and applying it to an ADC.
Oh, absolutely. And I think once we get to that position, then all we really need to think about is how we're attaching to the molecule. So we can target different DARs. We need to ensure that antibody drug conjugate as a whole, the biophysical properties are correct, and we're not just throwing as many drugs onto this antibody as possible and hoping that it does the job we want. We really need to understand exactly the PK profiles of these antibodies. And we're getting there now. We definitely are. There's fantastic conjugation techniques out there that give us homogeneity, give us slight selectivity. we can build them in. But I think we're missing something as well. We come back to the linker again. We can build in branch linkers. If we can build in the right branch linker with a single conjugation position, we completely minimize the biophysical property or biophysical effect that might have on the antibody. And we're just worried about the physical chemical properties of the linker itself. But we can modulate this with hydrophilic groups, or increasing hydrophobicity, or minimizing the amount of DAR that we have. So we're actually in a really nice position medicinally or medicinal chemically wise to be able to really understand those linker payloads and really understand how we can get a branched linker with a dual payload onto an antibody and minimize the biophysical properties of it.
And I think as we start to understand better the nature of that hydrophilicity, and how that affects the overall properties of the molecule, building that into our earlier screening cascades as we develop these molecules. So hydrophilic ion chromatography, the ways of assessing some of the, I guess, the in vitro DMPK. It's not something we've really thought about with ADCs. Now, clearly, the traditional assays we use in medicinal chemistry don't really apply for these molecules, but understanding those DMPK profiles, I think, is something that we're now moving much more towards.
Yeah, absolutely. We need to understand the metabolic profile of all of these molecules going forward, and we can do it. It's more in its infancy on the large molecule side of things. We can do it by affinity-based ELISAs, or we can do it by mass spec to a certain extent. The small molecule aspect of it, actually, we're really developed in. We can look at really high quality mass spec images. We can understand the metabolic profile really in depth. It's just bringing the two worlds together and making sure that both the large molecule and the small molecule world are bouncing off each other and being able to solve each other's problems. And I think we're starting to get there now. You can really start to see it build.
It feels like there's a momentum building. I guess the one bit we've not touched about, we talk about antibody drug conjugates. We've not really talked about the antibody bit, because it feels that you're with the evolution of aptamers, affimers, nanobodies, even like peptide drug conjugates. We're actually starting to move away from just, we'll take an antibody as the targeting mechanism. And it feels like there's a whole new range of worlds out there of different things that we can conjugate our linker and our payloads to. That gives a whole new set of properties that might be easier to deliver.
Yeah, absolutely. I mean, we always have to make it with that balance of, if we do use a smaller entity or a fragment antibody, we are going to affect the half-life, but we minimize all the complexity that you get with an antibody. We're not going to hit the same ADCC, ADCP pathways that we would do, or the CDC pathways. We've got an antibody itself is quite a complicated molecule, and there's so many functionalities that we don't necessarily need within an ADC, and we can pull it down to a purely delivery system with something like an antibody or an affimer or a bicycle or whatever is out there at the moment. And there's so much out there that actually it might well be the next step that we take into that field.
And I think the other thing that's out there that, again, we've perhaps overlooked somewhat through these conversations is most of the ADCs are in the oncology space. How long before we see an ADC in the non-oncology space?
Yeah, I don't think it's going to be too long, particularly with immuno-oncology and the immunology space particular. I think we mentioned it, one of the first ones is using things like M1, M2 macrophages and using something that's able to modulate between the two of them as an entity. I think moving away from the cytotoxic mechanism of action really opens the doors into new modalities for new toxicity.
I think it's really exciting, isn't it? I think that inflammation and immunology space feels to be the next frontier in ADCs. But one of the things as an oncology medicinal chemist that's always fascinated me is why we can't get certain small molecules up into the brain, across the blood-brain barrier, and yet we can get antibody therapies into the brain. It still perplexes me how that happens, but surely that's an opportunity then for CNS-derived ADCs. If we can start to understand how an antibody crosses the blood-brain barrier, be it compromised in disease or whether there's some active uptake mechanism, that's whole new vistas in terms of cognitive decline or pain or other areas that we've traditionally never even thought about in ADC.
Yeah, it definitely is. And it's still a bit of a dark art in terms of how to get an antibody into the brain. But obviously with Lilly and other people developing these things, they do exist. And with the nanobodies that are coming through now, the smaller entity is able to cross the blood-brain barrier slightly easily. So it's a world that I'm not an expert in, but I can definitely understand where it's going to go and how it can be able to be pushed into.
It just feels we've had 20 years of developing traditional, if you like, ADCs. It just feels that there are so many new unexplored vistas out there. I think that ideas, as we've discussed with thinking about the linker technology, the payload technology, beyond ADCs with a traditional antibody, patient benefit,
Mhm.
toxicity, therapeutic window. There's so much to explore out there in the ADC world at the moment. I think it's going to be an exciting decade. I mean,
Yeah.
how long before we start to see these molecules coming through into
I
development?
think quite quickly, but looking at the way the clinical landscape is at the moment, I think we've got a good few coming through very quickly, and then the real development really starts to move forward, and I think that's where it's going to be exciting.
It's a really exciting time to be in the space, isn't it?
Absolutely.
Yeah. Let's see where we are 5 years from now.
Yeah, that's it.